the first thing first to what I want to chat about is what we're going to study. Okay, and those are going to be microbes. So what are they? Both prokaryotic and eukaryotic organisms that generally are unified by the fact that they are really, really small and cannot be seen with the naked eye, except for certain exceptions. Okay, so smallest viruses, the hepatitis D, it's really a small guy, something like 20 nanometers in diameter. Polioviruses of a similar size, maybe slightly larger. They not only, you not only can see them with the naked eye, you cannot see them with a regular light microscope. And then when you go up in size, you will encounter larger viruses still, cannot see with a light microscope, and then bacteria. Bacteria can be seen in a light microscope. In fact, there is a microbe, an environmental bacteria called Thiomargarita namibiensis, which is about 0.5 millimeter large in diameter. So you can see a single cell with your naked eye. It's quite interesting. It's the biggest bacteria in the world. Okay. Same goes for viruses. We'll talk about it. There are several types of viruses that are so big that they can be seen with a light microscope, and because of that, they were discovered about 15 years ago. Nobody actually realized that those were viruses because they were so humongous. And then there are other microbes, protozoans, fungi, algae. Usually, except for viruses, everything else can be seen with a light microscope. Okay, but the range of sizes is pretty significant, from tens of nanometers to, in fact, meters in case of helminths. Helminths, what are helminths? Worms, right. Helminths, surprisingly, are subject of microbiology too. Although they're definitely not micro, like the pork tapeworm that can reach 30, 40 feet long in a human body. But they are infectious, and that's what we're going to focus on. We're going to focus on mostly clinically relevant microbes. We're going to talk about some that have obviously no role in disease, but mostly clinically relevant microbes. So helminths can be seen with the naked eye, like this guy, okay? Now, types of microbial cells. Bacteria. You see different shapes of bacteria illustrated in this picture. They are prokaryotic. Okay, so they do not have what? Nucleus. Okay. What else they do not have? Say again? Organelles. Very good. They do not have membrane-bound organelles, like mitochondria or endoplasmic reticulum. Right? And most of them can be seen with a light microscope. Although in that picture here, except for probably spirillum, all other little images taken with electron microscopy. Archaea. I don't have illustration for archaeal cells in this slide, but they look a lot like bacteria. And for a long time, we had no idea that archaea are fundamentally different from bacterial cells. That makes sense? So they look almost, okay, why almost? They look exactly like bacteria. But they're very, very much different if you will dig into the smaller details in the cell. Algae. You can see the wonderful different types of algal unicellular structures here. Now, Algae are eukaryotes, first eukaryotes that we mentioned so far. Are they unicellular or multicellular? It can be both, right? So algal blooms that you can see in Lake Erie when all the runoff with fertilizer goes into the lake, that's unicellular, guys. But if you swim in the lake and there's some freaky little things touching your legs that scare the living daylights out of you, those are multicellular algae. Okay, they belong to the same large group of organisms, but they're structurally different. Protozoa. So what does zoa mean? Animal, right. So they are essentially very, very small animals, okay? Unicellular animals. And that's a unique thing about them. They are all unicellular. All protozoans are unicellular. They're eukaryotic, so they have nucleus. Do you know any protozoans? Amoeba. Very good. Yep. Amoeba, they live all outside, in the soil and water, can cause actually clinical disease. Fungi. Those are eukaryotes. And for a long time, we didn't know whether we should put them into the realm of plants or realm of animals. Turns out they represent a separate group of organisms. Now, fungi, well, unicellular. Does anyone do mushroom hunting? You appreciate they're not all unicellular, right? The tasty ones definitely aren't. Okay? But clinically important ones, unless we talk about toxicology and poisonous mushrooms, clinically relevant ones, the ones that cause diseases, like Candida albicans, that causes thrush, or trichophyton, that causes different forms of ringworm disease. Okay, they are unicellular fungi. Make sense? Okay. Helminths. They, of course, eukaryotes, and they multicellular. That's the only multicellular organism in that list. Okay. The only clinically relevant multicellular organism in that list. You can see Dracunculus medinensis here and Taenia saginata, the beef tapeworms, the illustrations. I have a special place in my heart for helminths. Not literally. Some dogs do, but not literally. Okay, so they're multicellular, but since they follow very intricate and fascinating patterns of transmission between humans and the natural hosts and environment, we talk about them in the course of microbiology. And finally, viruses. So in my list of personal preferences for microbes, viruses take number one place. I'm a virologist by training, and these creatures are acellular. They're not cells. Okay? Now, anyone took 1200 or 1510? Okay. Good. Criteria for living things. What are living things? What do they have to have? They have to use and kind of convert energy from one form to another, huh? You've got to have metabolism, something else. They have to reproduce. Well, viruses reproduce, guess what? And metabolism is a slightly different story. They don't have their own metabolism. What is, yes? Yes. All living things are cellular. So viruses are acellular. So they're not living, right? Obviously, when I ask you a question in that tone, It's going to be a tricky question. So if I put the virus right here, is it going to multiply? What do you think? No. It's just sitting there. Now, anyone's gardening? Plants? Okay, good. Take seeds. Take seeds and put them right here. What's going to happen to them? Nothing. The plants, are they going to grow? So, seeds, are they alive? Well, they don't reproduce unless they don't produce the actual organism unless they plant it. So unless virus is planted into the cell, it's not going to reproduce. You see? So that more and more virologists actually think that we have to talk about two forms of viruses. The particle itself, which is definitely not living, and the cell that's infected with that particle. Okay? So that's a completely different story, and we'll talk about that. But I just want to plant that, sorry for that pun, plant a seed of doubt, seed of questioning in you. Don't hesitate to question me, things that I show you here, okay? Now, what are those? So, I didn't mention, so this is protozoan called Giardia. I like the name of the disease that it causes. The disease is called beaver fever. You get it from drinking from like streams and creeks. That's the fungus, Candida albicans, one that causes thrush and candidiasis. We talked about these two guys, the Tinea saginata and Proculculus medinensis. And that actually is a person who takes the little stick and rolls the dracunculus on that stick, pulling it out from the leg. Pretty boring and time-consuming process, and you cannot break it. Because if you break it, then the worm dies and your immune system starts to destroy it and it hurts. And those are viruses. So you see coronaviruses cause diseases like SARS, influenza, probably what I have now, the residual respiratory infection, and Ebola virus. We'll talk about the infection, we'll talk about the epidemics. I call it a much ado about nothing virus. Yeah, we'll talk about numbers and how dangerous it is and everything else. Now, there are branches of microbiology, like bacteriology, studies bacteria, okay, and also archaea. Phycology studies algae. Protozoology studies protozoa. Mycology studies fungi. Helminthology, obviously helminths, and virology, viruses. So except for phycology and mycology, everything else is pretty obvious, right? Yes. Mycology does, I almost said mushrooms. Well, mushrooms too, but fungi. Okay, so any form of fungus, whether it's yeast, okay, or molds. How important the microbes are. It's kind of a redundant question. Do you like beer? There you go. That's your fungus. Anyone likes wine? You know which part? So in France, people who work on the microbes inhabiting the surface of grapes, they get a lot of money from government because that's critically important for wine production. Okay, so those sets of bacteria and fungi that ferment. Nail polish? Well, I suppose. Acetone? Yes, acetone-based nail polish. Well, thank microbes for acetone. It comes from fermentation. Bread, cheese, okay? What else? I cannot exaggerate the importance of microbes. They are actually everywhere. And 2 weeks from now, you're going to run around the campus sampling whatever you want. The results are going to be surprisingly boring, actually. I don't want to spoil it, but they are everywhere. I don't think there is a single sterile surface on Earth, unless you're going to cut the person open and sample, I don't know, brain. But outside, they are everywhere. Like tar pits in La Brea in California, near Los Angeles. Come on, it's a melting tar. There are microbes in that tar. Okay. You see those corpses? Animals? Without microbes, we would be probably neck deep in dead stuff. They are major decomposers. Bacteria and fungi. Okay. Industrial applications, well, we just covered them. Not all, no. Do you like your water clean, the tap water? Well, think microbes. The huge microbial communities are designed specifically to clean water on the water treatment stations. So we cannot overestimate the importance. And of course, health. Clinically important microbes. Well, let's see. Heart disease, okay? Is that infectious? Some of it is. It turns out some of it can be due to the infections. Stroke, again, vascular disease can be associated with microbial infections. Chronic obstructive pulmonary disease, well, it's mostly smoking. Microbes probably contribute, but not that much. Low respiratory infection, read pneumonia, stuff like that. Trachea, respiratory infections, HIV, diarrhea. So that block right here is all infections. Those are causes of death, okay? Now, death is important. What's worse than death? Yes, bingo. Not like me. I hope I'll be fine. But consider this. To have that kind of a problem for years. It hurts. It really hurts. And it's bad, but it's not worse that can happen. Sorry. Think about malaria. Every 3 days, the patient is going to have chills, fever, bone aches. You're not going to work with that. And if you have it like every 3 days, your life sucks. You can't work, you know? So that is worse than death in some way. Okay, you're going to live like that for years and years and years. The quality of life will be impaired. And that's the situation where diseases, infectious diseases, I would say they have the first place in terms of how they affect the humanity. According to estimations, about 1/3 of the world population is infected with helminths, different helminths. And it all affects their lifestyle. Now, any questions so far? So please know the branches. Sorry. And know the details about microbes that we talked about. Like if I ask you, which of these microbes is prokaryote? And I tell you, fungus, algae, virus, and bacteria. What would you say? Bacteria, yes. Virus doesn't cut it because it's not a cellular thing, right? So know these characteristics and know the branches of microbiology that address different microbes. Right? Now, we have to name them. Taxonomy. The first taxonomist was Carl Linnaeus, a Swedish guy. And he pretty much invented the modern way of naming things. Okay? So it's a two-word names. Of course, he didn't talk about microbes at his time. It was like 18th century. He talked about big animals. But that system was accepted worldwide and is still alive and well. Two words. The first word is for genus. Second is for species. Now, a little reminder about the classification. So species, can you give me an example of species? Don't have to be microbe. Yeah, animal. Give me a species. Felines will be not species. It's broader. It's even broader than that. Even broader. You go you go up. You're going from very narrow, very broad. Any see 18 of the particular species. Huh? Homo sapiens, yeah, we species. Essentially species, right? Homo sapiens. Okay? Now, which genus do we belong to? Homo, okay, so genus. Okay. What is above genus? I'm not talking about us anymore, because I don't remember. It's not yet. Family. Family, then, anyone knows? Order. Then usually class. Very good. But class, sometimes it's a type. Then it depends for different animals. It can be class that belongs directly. It can be a phylum, may not be a phylum. So usually class, type, phylum, kingdom. Okay? So we go from very, very narrow to very, very big, very broad. Okay? So felines that Jeff mentioned would be a family. Okay? Animalia that Brianna mentioned would be a kingdom. Mammals that Jamie mentioned would be a class. Okay? So same classification can be done for bacteria. With viruses, it's slightly different. They have one word name. We'll chat a little bit about virus nomenclature. It's a pain in the ass. But how do we relate things? There were different ways to classify organisms, and the one culmination of that taxonomy, of systematics, was when Robert Whittaker in 1969 proposed his system of 5 kingdoms that would take into account evolutionary relationships, mostly, okay? Evolutionary proximity of different life forms. So it would be animalia, animals, plants, fungi, Monera, and protists. Okay, so 5 of them. So for example, Monera would include all bacteria and archaea. Protists would include amoeba, for example. Animalia, fungi, and plants are pretty obvious, right? So there were a lot of criteria. Structural similarity, how they look. Okay? how they related to each other in terms of evolution. But what is important is 1969. So he already came up with that idea. Around that time, I think it was late 60s, maybe early 70s, the biologist named Carl Woese was working at the University of Chicago, and his work was dedicated to the genetic classification of microorganisms. So we all know that living things have genome, right? The genome encodes all information about any living thing. And there are certain parts that are more variable, like very, very variable. They can vary between different people, okay? And then some parts that are more similar. They may be 100% conserved. What conserved means? Like if something is conserved in the genome. Not necessarily not active. When you say somebody is a conservative, don't like change. So conserved parts of the genome, they don't change much in terms of one species. You jump to another species, there are minimal changes. So it turns out, the more changes there are, the more difference there is between conserved elements of the genome, the farther those species are from one another. Does that make sense? Like if you would compare, so there's a 16S ribosomal RNA gene, specific gene, that every bacteria has. Every, not kidding. Bacteria, mammals, all organisms. It's a fundamental gene, okay? And if you would start comparing that gene among the same species of bacteria, the gene is identical. You compare it among humans, the gene's identical. You're comparing species, genes are different. Does that make sense? I'm trying to find an analogy. I'm a sucker for analogies. It's similar to what we talked about in terms of the Whittaker system. Like you compare all cats, they all have a lot of common features, okay? And if you compare just cats, they look identical except for the color, pretty much. You compare cats and, I don't know, bobcats or lynx, more differences, okay? They're still similar. You go to dogs, well, pretty much the common thing is that they all have 4 legs, okay? The same goes for that gene. The more distant species you look at, the more different the gene is going to be. And vice versa, if you compare 2 genes of 2 species, and they are very, very different, you can say, oh, those species are very far apart on the evolutionary tree. Does that make sense? Like, especially with bacteria, they all look the same, come on. You look at this, it's like little rod, and you look at this, it's little rod, they look the same. How do you tell if they evolutionary close? You have to look at the genome. And that's what Carl Woese did, was looking at the genomes. Well, it was his grad students and postdocs who were doing the job. He was a boss. Anyway, he was studying it. It was good work. He didn't discover something like exceptional at the time. And once he asked his colleague in the same department for the sample of microbe that colleague was working on. It was some methanogenic microbe. And everybody thought it was a bacteria. So he ran the 16S RNA analysis. He looked at 16S RNA of that methanogenic microbe, looked at 16S RNA of bacteria. And there was like nothing in common. And I thought, oh, come on, that's not true. That's some mistake. So his postdocs and grad students, poor fellas, did that run over again. Same result. So it turns out, among those microscopic creatures, there's an entirely different domain of life, okay, that we call archaea. So on the genetic level, according to the Rose Fock system, there are 3 domains of life: bacteria, archaea, and eukarya. And that tree here is called phylogenetic tree. Okay? Let's name it. It's phylogenetic, which means it shows the relationships between different types of microbes or living things, microbes, protozoa, whatever, okay, based on their genetic similarity, exclusively genetic similarity. Okay, 2 organisms may look absolutely different. You look at the genes and say they related, closer than you think. Does that make sense? Yes. Where's that? Oh, thank you for spotting it. My bad. You can. Ah, prokaryotes. Yes. Yes. Yeah, but I understand the confusion and I should correct it. Thank you for saying that it's correct, but I understand the confusion. So you have 3 domains of life: bacteria, archaea, and eukarya. These 2 do not have nucleus. Does that make sense? Okay? Now, a lot of scientists say we shouldn't use prokaryotes anymore because it's kind of not actually relevant. I find it very convenient to use, honestly, because we talk about fundamental structural feature. So I'm going to use it throughout. Okay, when I say prokaryotes, it means it can be either bacteria or archaea. Now, if we would look at the phylogenetic tree right here, you will see that there was some sort of the common ancestor. That common ancestor gave rise to 2 branches initially: bacteria and the common ancestor of archaea and eukarya. So essentially, although structurally archaea and bacteria look very much the same, genetically, functionally, archaea are closer to us than to bacteria. Does that make sense? They look nothing like eukarya, but genetically turns out, and functionally, they're closer to us. They have a lot of similarities with eukaryotes. Okay? Now, we're going to chat about the history, but we're going to do it after the break. Well, actually, after the break, we're going to do the lab. And then we're going to chat about history, okay?